back with another passage. You already know how it goes. Eric here. For those of you guys that don't know me, my name is Eric, founder of Eric Med, founder of MCAT University, and it is my mission to make sure that this MCAT is as easy as possible for you guys, especially in the CP and BB sections, because that is where I specialize in. Okay, I got a 132 in both sections this past January on the MCAT, so I know what happens here. I've also tutored plenty of premeds to hit their target score. Okay, so I'm going to be breaking down this passage for you guys today. All right, this is passage number 2 on the Blueprint FL exam. It's completely free. You just sign up for a Blueprint account and they give you this for free. Now, I'm going to show you guys exactly how to read the passage, how to highlight, how to pick the best answer, how to make this whole thing make sense, all this gibberish here. Okay. I'm going to show you guys how to choose like the best answer choice. Okay, I'm going to show you guys how to do everything in these passage breakdowns. As always, guys, go ahead and do the passage first. All right, read through this passage first and answer the questions on your own first, and then hear me break it down. Okay, if you need to, pause it whenever you need to. All right, this is the first question. This is the second, third, what was that? Fourth, and fifth. Okay, cool. So pause it whenever you need to, guys. Write down your answers and check to see where you messed up, what you got right, what you did right, what you did wrong, all that stuff. I'm gonna break it down right now. Here we go. Let me sip in this water first. Okay. Giardiasis. All right. Caused by the intestinal flagellum protozoa Giardia lamblia is a common intestinal disease. Okay. Protozoa. You guys should know what protozoas are. They're single-celled organisms. Okay. Giardia are unique in their possession of 2 nuclei that are similar in appearance. Okay. Remember, guys, whenever they say that something is unique, whenever they say something, oh, this protein is different than all the other proteins because it has the blank, blank, blank function. Okay, whenever they say something that's unique or different than all the others, you highlight that. You highlight that right away, because more than likely the question is going to be about that specific special thing that the bacteria or protein has, okay? So what am I highlighting? I'm highlighting their uniqueness in that they possess 2 nuclei that are similar in appearance, DNA content, and transcription. Usually, G. lamblia are transmitted by contaminated water with cysts. G. lamblia have 7 complex genotypes, but only 2 genotypes cause human infections. The illness is transmitted by differentiation into infective cysts. 3 known cyst wall proteins are highly synthesized in a concerted manner during differentiation into cysts. Okay, so it's transmitted by differentiation into infective cysts. Okay, it's the cyst wall proteins that are synthesized in a manner during differentiation into cysts. Okay. Simple paragraph here. We got a disease, okay, and then how does it transmit the disease to other, species, hosts, whatever. It does this by differentiating into cysts. It's all about the cyst. Okay, cool. Encystation specific vesicles, ESVs, I'm going to highlight this, give the organism the ability to modify or partition cyst wall cargo during secretory transport. These organelles aid the accumulation and maturation of cyst wall material composed of at least 3 cyst wall proteins complex with beta-N-acetyl D-galactosamine polymer. Wow. Okay. CWM protein is eventually deposited in the plasma membrane of the host intestinal cells. So this CWM needs to aid, the organelles aid the accumulation and maturation of cyst wall material of these cyst wall proteins coupled with this polymer, and then that is deposited into the host intestinal cell. All right, crazy stuff going on here. Trafficking of mature CWM from ESVs to the cell surface where it forms the cyst wall is rapid. During cyst formation, a round of DNA replication occurs. All right. Then nuclear replication takes place, okay, with the DNA divided equally among the resulting 4 nuclei, followed by a second round of DNA replication. Okay, we have a lot going on here, guys. A lot going on here. Okay, here's the figure. Okay, don't bother, in analyzing this figure. Only analyze a figure when the question asks for it. If you spend time trying to look at it, you're going to waste your time and you're going to forget what you just read a little bit. Okay, you want to read this and kind of have some of this in the back of your mind. All right, the longer you go, the more likely you're going to forget. You can read the caption though. All right. Regulation of CWP abundance within 2-hour transitions during the 14-hour encetation time course. H pi equals hours post initiation of encetations. N equals total protein in the sample. At any given time throughout the parasite's lifespan, only one CW gene is expressed. During an infection, however, the particular CW gene that is transcribed regularly alternates in a process termed antigenic variation. Okay. Both expressed and silent CWP genes are located in the nuclear periphery, but in different specific regions. In expressed CWP genes, promoter histone H4 is acetylated on arginine 6 and trimethylated on arginine 2. In contrast, silent CWP genes include a trimethylated histone H4 at arginine 6. All right. Whenever they go into these details, like this gets acetylated here at this one and this, these details, it's okay. You don't need to highlight them. You don't need to really remember them. All right. Because if the question asks for it, then you can go ahead quickly, bam. They're not going to ask you something complicated about this. All right. So this one you kind of speed through with your reading. All right. So if I was reading it, I'd be like, in expressed CWP genes, promoter histone H4 is acetylated arginine 6 and trimethylated arginine 2. And in contrast, silent CWP genes include a trimethylated histone H4 at arginine 6. All right, that's how I'd read. I'd read a little quicker this. I don't want to waste my time on it. During the homologous recombination of cis nuclei, type 2 isomerases assist in chromosome replication and tissue development for differentiating to dormant cis. Okay, remember, all about those cis. All right, we're differentiating to cis. And this type 2, this helps. So that's pretty important, this isomerase. In addition, the levels of cis formation and the CWP 1 to 3 gene expression are increased by topo O2 overexpression. Cool. Topo 2 is good. This is good. All right. Topo 2 overexpression is good. Cool. Long. And actually, no, not too long, but it's just on the long side, this passage. Okay. We have intestinal disease. All right. We have the vesicles that help transport all the stuff. All right. We have the wall proteins that are deposited in the host intestinal cells. We get that cyst. It's all about that cyst. I said cyst a million times, but we get that cyst. All right. And then they tell us how this gene process works. Okay. All right. And then they tell us isomerases helps a lot. That this is good. It's topo 2 overexpression is good. Honestly, this passage is all over the place. If I was reading this as a scientific reviewer, peer reviewer of scientific papers, I'd be like, dude, you got to organize this better, man. Like, that's what I would say, but it is what it is. Okay. Do I feel amazingly confident about this passage? No. Okay. There are easier passages out there. Do I feel good about it? Yeah, I feel good. Okay. What is the expected ploidy of a cyst that has completed an encetation? I can never say that right. Okay. You should know what ploidy is. Ploidy refers to the set number of chromosomes. Okay. So like, let me go to the whiteboard here in case you guys don't know what this is. Okay. So let's say if you have 2N, okay, you have a diploid. You have two sets of N, meaning number of chromosomes there. You have two sets of chromosomes. All right, and this is in a nuclei. Circular refers to nuclei. If you have 4N, you have 4 sets of chromosomes in one nuclei. All right. If this is an organism with 2 nuclei, you have 6 sets of chromosomes in that organism. Okay. I'm sure you guys kind of know that about from your constant review, but if you haven't, that's pretty important. Ploidy is important. Okay. So, what is the expected ploidy of a cyst that's completed an end citation? Well, if you get a question like this, specific, you could reread it. It's okay. All right. During cyst formation, a round of DNA replication occurs, then nuclear replication takes place, with the DNA divided equally among the resulting 4 nuclei, followed by a second round of DNA replication. Okay. Remember, all right, this protozoa has 2 nuclei. All right, and I think it tells us, all right, right here. Giardia are unique. Remember, guys, unique. Or when they tell you, hey, this protein is different than this other protein, is because blah, blah, blah. I promise you it's going to be important. Highlight it. The question is going to ask for it. And if I did not highlight this, I probably would have forgot about that little detail. Okay, so MCAT tip right there. Make sure you write that down, what I just told you, because that's freaking important. Okay, that's going to give you some points. All right, that's AMC logic for you right there. You're getting that test maker's head. I know that this test maker is going to include something like this. I know it. Okay, that's how you guys should think. All right. So let's do this. So we have 2N, 4N. Okay. All right. We have 2 nuclei that are similar in appearance. All right. Protozoa, you should notice from your content review too that they are diploid. Okay. They're diploid. And they mentioned that they were mature a bunch of times, I'm pretty sure. So you should assume that they're diploid. And they have 2 nuclei. Okay. So this is what we got starting off here. So what happens? A round of DNA replication occurs. All right, if we have a round of DNA replication, this is going to become 4N. Okay, 4N. This is going to become 4N. We replicated the DNA. Okay. Then what happens? Then nuclear replication takes place with the DNA divided equally among the resulting 4 nuclei. Wow, that's okay. So we have the nuclear replication. Okay, we're going to replicate our nuclei here, and it's going to be divided equally among the 4 resulting nuclei. So we're going to result in 4 nuclei. All right. So 4 nuclei. We have 1 nuclei here, another nuclei here, another nuclei here, 4th nuclei here. Okay. And we are dividing the DNA equally among the resulting 4 nuclei. So this is our total DNA here. Our total DNA is like AN. Okay. A sets of chromosomes. All right. We have to divide this equally among the 4 nuclei. So A sets of chromosomes in 4 different nuclei means 2N per nuclei. Okay, we have 8 in total here, 8 sets here, but we need to divide it equally among the 4 nuclei. So we have 2N here, 2N here, 2N here, 2N here. Okay, I'm gonna say it again. We divided the DNA equally among the 4 nuclei. Okay. Next, what happens next? I think they replicate another time. Okay, followed by a second round of DNA replication. All right, DNA replicating, not nuclei, DNA is replicating. So this is going to be 4N. This one's going to replicate to 4N. This one is going to replicate to 4N. This one's going to replicate to 4N. This is what we got here. And that's it, right? Yeah, that's it. So they asked for what is the expected ploidy of a cyst has completed. So this is after everything. It completes this end citation process. We have 4N, 8N, 12N, 16N in total. All right. The answer is D, 16N. All right. That question's a little annoying because you have to see nuclear and then DNA divided equally. That's a little annoying, but the answer is D. The functions of the ESVs of Giardia most closely resemble what cell organelle? ECVs are end citation specific vesicles. All right, that function in secretory transport. All right, That's pretty easy, guys. Lysosomes, that has nothing to do with this. Lysosomes break down stuff. Okay, they have those enzymes in there. It's pretty acidic. They digest, they break down. So the answer is not A. Golgi apparatus. I like this answer a lot. Okay, because what does a Golgi do? I mean, if you guys are very, very good with this, then you can skip this part here. Just hit the next part. But I'll explain it to you real quick here about Golgi and secretory transport. Okay, so here is the cell. Here, I'll bring it over here. Here is the cell. Here's a nucleus. I'll put an N here for nucleus. Here is the endoplasmic reticulum, let's say. And then from the endoplasmic reticulum, we have the Golgi. All right, and this is the Golgi here. All right. When we want to secrete something from the cell, let's say a protein that we just made, all right. Here is a, can I do a different color here? Oh, never mind, guess not. Okay, here's a ribosome. Okay, this is a ribosome here. This is the large and the small unit. Let's say this ribosome just made a protein, and I'll make this protein a triangle. Okay, this ribosome, the protein it just made, oh my bad. Okay, so this ribosome, this is not systolic. This should be located on the endoplasmic reticulum. Okay, so this is our ribosome here. All right, forget this. Don't look at this. Okay, here's our ribosome studded on the endoplasmic reticulum. It makes a protein. Okay, this protein is here in the endoplasmic reticulum. From the endoplasmic reticulum, the protein goes to the Golgi. Here's the protein, the Golgi. The Golgi then does some modifications, whatever, but then it packages it into a vesicle. Okay, and this vesicle has two fates. This vesicle with the protein in it can be embedded in the plasma membrane and stay there in the plasma membrane, or it can be secreted. Okay, and this secreted protein can then travel whatever it needs to. All right, maybe it's insulin that travels in the blood to a target cell or something like that. So that's a secretory transport. All right, and the Golgi does that. The Golgi works in that. Nucleolus, no. Centriole, no. Answer is B. Given passage information, Giardia most likely transports which of the following proteins external to the organism itself? Okay, CWP3, that is the wall, right? Yeah, that's the cyst wall protein complex with this polymer here. And this cyst wall protein is eventually deposited in the plasma membrane of the host intestinal cell. So that means that this protein had to be secreted from the cell. It had to be secreted from the vesicle here. Okay, so this vesicle transports the CWP protein, all right, to the plasma membrane and ejects that CWP3, that CW protein, okay, and that protein goes and then deposits into the host cell, into another cell. All right, so this is what it does. So that protein, let's say this one actually is CWP3. That goes, and then let's say this is a host cell. That goes, and then it gets deposited into the host cell plasma membrane. So this had to be external. This is external. All right. I don't know why they use external, that word. They could have used another word, in my opinion, but that's what that means. So this makes sense here. Histone H4, no, that's in the DNA. That's in the chromatin. That has nothing to do with being secreted or anything like that. This polymer here, oh, this is coupled with the CWP. So there is a chance that this can be ejected as well. ESV, that's the whole process. So no, that's wrong. That's the whole overarching process. Okay. Now, here's CWP3. Now, which one is it? This one or this one? Okay, let me sip on my water again. We have to pick the better answer choice. The better answer choice is CWP3. Why? Why is that the better answer choice? Well, there's more evidence to this. They talk about this more. Okay, remember they talk about C-wall protein is then deposited into plasma membrane. And then there's traffic from CWM to the cell surface where it forms a cell cyst. It's rapid. There's more evidence to this. And yeah, there's more evidence to this. This one, they talked about it just for a second. They just said, oh, it's complex with this. We don't know if this one gets injected into the host cell protein or not, the host cell membrane or not. I'm sorry. We don't know if this gets incorporated into the plasma membrane of the host cell. We know CWP does. We know a C-wall protein does, but we don't know if this one does. So this one's more supported. This one makes sense more. So I'm going to go with this one. With which of the following do the arginine side chains found on histone proteins most likely interact? Arginine is positive. It's going to react with something negative. Not this, not this. Phosphate. Phosphate has that negative. The data in the passage suggests that which time period during end-citation is the level of protein dropping most quickly? Guys, this is so easy. Okay, look at the figure. We want something that drops most quickly. This means up, not change, down. Down regulation, up regulation, not change. Down regulation means that the protein level is dropping. And so we look which one has dark light gray here. Okay, this is like 2. This is a whole 15. This is like a 3, 0, 5, 5. This is obviously the biggest drop here. So the answer is 4 to 6. And that's it, guys. And then we'll do this passage next video. All right. If you guys are interested in working one-to-one with me inside MCAT University, go ahead, guys, and go to the comment section and click book a interview, and we're going to see if we're a good fit, and we'll work together. If you guys want to see the answers to these questions, here they are, all right? Let's see if we got them all right, actually. Should have done that earlier before I plugged in MCAT University. Okay, 16N, that's one we chose. We got that right. Here is the explanation if you guys need. That was a hard one. Wow, that was a really hard one. All right. Golgi. Explanation. CWP3. Okay. And the phosphate groups. And the 4 to 6. All right. If you guys are interested in working one-on-one inside MCAT University, guys, applications are now open. So go ahead, go to the comment section, book an interview. We'll see if we're a good fit. Comment down below what you guys think. You know, if I messed up anywhere, comment that down below again. Like, yeah, if I messed up anywhere, comment that down below and, you know, call me out on it. Okay, you guys should look at what I say and hear what I say and be like, I don't know, Eric, maybe this, that. You know, be a reviewer. We review this stuff together. We work together and we, you know, go and we get that MCAT score. That's what we do. See you guys in the next video. Peace.